Um, so finishing the lecture from last week about muscle tissue and muscle system. So this is where, of course, we were looking at all of the things that it takes to actually make a muscle contract. And we were looking at the smallest pieces, the actin and the myosin arranged in a sarcomere. Okay. This piece here is actually more of a big picture view. So recall, towards the end of lecture last week, we were talking about muscle fibers firing. So refresher here, muscle fiber, same thing as a muscle cell, just different name. When a muscle fiber fires, you can say it also as when a muscle fiber contracts. This is the same thing. It does this because it is receiving an electrical signal, an action potential from the nervous system. Specifically, that signal travels down what is called a motor neuron. We're going to look at motor neurons today, so that part will make a little bit more sense. The motor neuron is the nerve cell that signal travels down from the nervous system, from the brain, from the spinal cord. So the motor neuron can then communicate and tell muscle fibers to work. A motor neuron plus all of the muscle fibers that motor neuron talks to is called a motor unit. A motor unit is one motor neuron plus all the muscle fibers that motor neuron can innervate, can make contract. Does that sound a little familiar from last week? A motor neuron can be something that is very, very small. A motor unit can be very, very small, where we have one motor neuron that is just controlling one single muscle fiber. This would be in areas of the body where we make really fine, really precise movements, like in our eye, for example. Or a motor unit can be very large, where we have, again, still one motor neuron that tells many, many muscle fibers to work. These would be our large muscles, like some of the larger muscles, skeletal muscle in our lower body. So let's think about a large muscle, just to kind of get a more specific example. Let's think about a large muscle, say something in the lower body, in the legs. When we are running, for example, we're using a lot of different muscles, lots of different muscles in the legs. These motor units are going to recruit muscle fibers that are sprinkled all throughout the muscle. So it's not like only one tiny little piece of the muscle, only these muscle fibers are working at a time. The motor unit is going to recruit muscle fibers all throughout the muscle, and they're scattered all throughout. These muscle fibers, we have different types of. So different types of muscle fiber, and each different type of muscle fiber has a particular quality to it. And it is typical for people to have an arrangement depending on genetics and activities or sports that they do, a different makeup of how much of what type of muscle fiber you have. So starting off here, we'll talk about fast fibers. If you've had other kin classes, you've probably heard of these different types of muscle fibers before. There are some different names. So fast fibers can also be called type 2B or just type 2. Fast twitch, glycolytic fibers, fast fatigue fibers, although I never really heard anyone refer to them as fast fatigue fibers, or white muscle fibers. So your fast fibers, I tend to call it fast twitch or type 2 muscle fibers. These muscle fibers, as the name implies, are really good at contracting very, very quickly. They're also a lot better than other muscle fiber types at creating forceful, powerful contractions. forceful, powerful contractions. So if we're thinking about like elite athletes that have lots of type 2 muscle fibers, these are going to be anything that involves explosive, very quick, maximal type of movements. So sprinters, Olympic lifters, those type of athletes, very, very fast. These muscle fibers contract very quickly. They're also larger in diameter. Okay, if you think of in your mind the body type of a sprinter and how muscular and large muscles a sprinter has compared to an Olympic marathon runner, both elite, both best in their class. But the marathon runner has a very different body type, right? The muscles, although very highly trained, are much smaller. That's because the sprinter is going to have much more fast fibers, which are able to increase in diameter. So these muscle fibers are more powerful. On the flip side, though, because they are fast fibers, they also fatigue quicker. This is why these types of explosive, powerful exercises aren't sustained for a long time. Olympic lifting, you're doing a certain lift, takes a few seconds. Sprinting, depending on the length of the sprint, also we're looking at under a minute, under 30 seconds, under 20 seconds, because these muscle fibers fatigue very quickly. Because they fatigue quickly, and we're looking at short bursts of energy, we're looking at mainly anaerobic metabolism. Anaerobic metabolism means that we are able to supply these muscle fibers with energy without using oxygen. So fast twitch fibers, big, powerful, explosive movements, but fatigue fast. Slow fibers, also known as type 1, slow twitch, slow oxidative, or red muscle fibers. These are kind of like the opposite. So as the name implies, they are going to contract relatively slower, so slower than the fast twitch muscles. They do not get as large. So again, in your mind, thinking of an Olympic sprinter versus an Olympic marathon runner, the Olympic marathon runner who is also extremely highly trained, the muscles they are not as large physically in diameter as the sprinter, because these muscle fibers simply don't grow in diameter as much as the type 2 ones do. On the flip side, though, these muscle fibers are really great at resisting fatigue, so they are able to last a lot longer and fatigue slowly. Because of that, we dip into aerobic metabolism. Aerobic metabolism is where we are now utilizing oxygen in order to produce energy, ATP. There is a tremendous amount that could be said about that, which we simply don't have time for, so we're just going to note that. When we start to use oxygen in order to supply the body with energy, it takes more time. And so these types of activities are ones that last longer. These are also called the red muscle fibers because they contain a pigment, myoglobin, that gives it a red color. And then between type 1 and type 2 muscle fibers, we have intermediate fibers, which are just like the name implies, it's something in the middle. This is interesting to look at. So again, everyone is going to have all different types of muscle fibers. How much you actually have is dependent, again, upon your genetic makeup and also the activities that you do. So we do have an ability, a limited ability to a certain degree, but an ability to train and shift our muscle fibers to have more type 1 or type 2, depending on what activities we're doing. So if we were to look at, here's a world-class sprinter. The world-class sprinter is going to have a much higher percentage of fast twitch fibers because they're participating in a fast explosive type of exercise. An extreme endurance athlete or a marathon runner, as you would probably think, going to have a much higher percentage of the slow twitch fibers. And then as we get into the center would be different types of activities. Middle distance runner is going to have slightly more of slow twitch. Average active person is going to have about 50-50. And this one's interesting to me, and I love how they say average couch potato. Cute. Is going to have slightly more fast twitch fibers, which is an indication that once a person starts getting off the couch and training a bit and just has an active lifestyle, some of these muscle fiber types switch and turn into more slow twitch fibers. Okay, so when you exercise and you train a muscle, the muscle can increase in size. When a muscle increases in size, it increases in diameter. This means that the actual muscle fiber itself gets bigger, not the number of muscle fibers. We cannot increase the number of muscle fibers. We can just make the ones that we already have bigger. When we make the muscle fibers that we already have bigger, that is called hypertrophy. Any guesses as to what happens in the muscle fiber that makes it bigger?
I think I remember reading about this. Isn't it that the fiber gets damaged and then gets built back up?
Yes, that's true. That's part of the process of how it happens. But what is it that there is, say, more of that's making the diameter larger? This isn't anything that we've explicitly said, so I'm just wondering if anyone has guesses. But
Resistance?
No. I'm talking about, think about like the actual muscle cell, the muscle fiber, everything that goes into it. Something's got to be making it physically bigger, so we've got to have more of something.
Protein.
Okay. Is that what you're going to say? Okay. Yeah, good. Protein. Yeah. Yeah, mitochondria. Good. Okay, so so more mitochondria. So remember, mitochondria are the organelles that make ATP. So this is an adaptation to training. As we train and we exercise, we're able to increase the amount of mitochondria that we have in our muscle cells. This is great because if we have more mitochondria, we're able to make more ATP. More ATP, more energy available to the muscles, we're able to produce stronger muscle contractions for longer amounts of time. So it's a wonderful system. More glycolytic enzymes, larger glycogen reserves. So remember that we store, the stored form of glucose is glycogen. We store that in the muscles.
So if the greater concentration of mitochondria allows for stronger but also longer contractions, would it be, so if the average couch potato is in the middle, or not balanced, but almost,
yeah.
If they lean one way or another, wouldn't technically the mitochondria be, wouldn't they be producing like the same rate? Is that right?
You had me up until the end. You lost me a little bit.
Basically, like if you train for like short bursts versus endurance, which one will build more mitochondria faster? Is what my question is.
Oh, which muscle fiber type has a higher affinity to building more mitochondria?
Yes.
Mitochondrial density is what we call the measurement of how much mitochondria any particular cell has. That's a really specific question, one that I haven't looked at in that great of a detail in a while. I don't know if one muscle fiber or the other has a higher affinity towards increasing mitochondrial density because they're both going to need it, right? So that's something that I'd have to look up and see. Certainly, type 2 muscle fibers are able to produce stronger contractions, but that doesn't mean that a person with training that has more type 1 muscle fibers doesn't also see an increase in mitochondrial density, because they absolutely do. Yeah, you're going to see an increase with training regardless, but it's a good question. More myofibrils. Myofibrils, these are these cylinders that run the length of the muscle fiber. The myofibrils are where the sarcomeres are stacked together. So although we cannot increase the number of muscle fibers, we can increase the density of myofibrils within those muscle fibers. And if we're increasing myofibrils, then we can increase actin and myosin. And at the very smallest piece of it, if we have more actin and myosin, we have more cross bridges. More cross bridges gives us a more powerful contraction. That was actually kind of a lot to take in. Questions on that piece? Does that make sense? Okay. Increased aerobic endurance is the result of an increase in mitochondria numbers, like we were just talking about. So increased aerobic endurance would be looking at more like type 1 type muscle fibers. So we're still seeing an increase in mitochondrial numbers and capillary density around the muscle. So what this means is with aerobic training, so exercises that force us to move our bodies more than 2 minutes and we deplete all the other energy sources. With aerobic training, we see more mitochondria in the muscles and a higher capillary density. This means more blood flow to the muscles. We're able to get more oxygen and nutrients to the muscles, which is also a good thing. We get lots of stuff that we need to keep the muscles going. Those are adaptations that we see with training. So then if we're looking at an aerobic exercise or an endurance event, what is going to be the limitation on our bodies? So at what point do we say, I'm tired, I'm done with this aerobic exercise? What are going to be the limiting factors? Again, not something that we've specifically talked about, but if you have any thoughts or guesses.
Lactate threshold?
Yeah, that's definitely something we haven't talked about in here, but do we even have that up here? It's certainly a correct answer, but something that we look at more in other kin classes. Lactate threshold, the buildup of lactate is a byproduct of when we utilize our muscles. How much ATP we have available and creatine phosphate we have available. How much glycogen we have available for breakdown. Oh, there we go. I thought maybe I had some lactic acid. How much lactic acid buildup we're able to tolerate. And then it's the last one, something that you are already familiar with. Origin and insertion. This is the beginning point and end point of a muscle. So all muscles are attached, all skeletal muscles are attached to bone. Origin is the beginning point on the bone. Insertion is like the end point. For every muscle action, we have the main muscle that is moving, which is the agonist. And then we have an opposing muscle group is the antagonist. Then we also have helper muscles that help the main muscle move. That would be the synergist. So we're not ever really just doing like working one muscle by itself, because for the most part, when we're moving our bodies around, there's going to be multiple muscles that are helping in any particular muscle action. And then innervation, as you know, is the name of the nerve that tells that muscle to move. Okay, done with your favorite lecture. I know you're sad to see it go, but questions on that last bit? Okay, let me pull up the one from today. All right, so we are going to start the next section, which is looking at the nervous system. So we will do this in lab also. We're going to start this next week. The lecture that we're doing today is pretty much just looking at the basic components of the tissue that we see in the nervous system. So we're just going to be naming a lot of the pieces. No complex process that we're looking at today. Probably a lot of new words for you, but words you are also going to see in lab. So lots of repetition there and nothing terribly complicated. Okay, so the nervous system. We have talked about the nervous system a bit as we've been going through. but now it will get our main focus. The nervous system is how we receive information about the external environment and also what's going on inside of us. We receive information, we process that information, and then we get a chance to respond to it. And that's the nervous system. So the information that we receive is called sensory information. And there's a whole host of different types of sensory information that we receive. And we will take our time going through and talking about the different pieces of the sensory information that we receive. But essentially, sensory information comes in. It comes into the central nervous system, which is the brain and the spinal cord. We process that information, and then any response that we want to make about that sensory information then gets integrated into the response, which would either be a voluntary response, like the room getting very warm, and we receive that sensory information. We decide to take off our jacket, so we coordinate that response to our skeletal muscle, and we start removing layers. Or involuntary responses, which would be telling the body to start sweating. We don't have to consciously control, like, hey, body, start sweating. I'm too hot. So there's two divisions of the nervous system. We have the central nervous system, which is abbreviated CNS. The central nervous system is the brain and the spinal cord. The sensory information that we receive comes in through the other division, the peripheral nervous system. The peripheral nervous system is the communication with the rest of the body. So the central nervous system is hooked up to the peripheral nervous system. Within the peripheral nervous system, we have two subdivisions. The afferent division, which is the sensory input, and the efferent division, which is the motor output. You are going to see afferent and efferent being referenced a lot, so it's worth putting a star or making a note somewhere. Afferent equals sensory. Efferent equals motor. So the afferent division, the sensory division, is where all the peripheral information comes in, gets sent to the central nervous system. We process that, and then if we're going to make a response, then that response travels out through the efferent division, through the motor division, to then carry out whatever response we want to make about it. Did you have a question? Okay. All right, meet the neuron. A lot of these terms here are going to end up being terms on your list for labs, so really nice overlap here. A neuron is just the fancy name for a nerve cell. Just like muscle fiber is a fancy name for muscle cell, it's the same thing. What exactly is the neuron? All of this here, plus that little highway, not this. Does anyone know what they're showing us here? Yeah, awesome. That's a muscle fiber. So that's not the neuron. This is actually a neuron talking to a muscle fiber. But all these little lines, all of that is the neuron. So we're just going to go through and name some pieces here. The cell body is this larger expanded portion here. This is where we have the nucleus and the other organelles. The cell body. One thing that we do not have in neurons that some other cells do are centrioles. That might sound a little familiar from beginning of the semester when we looked at cells. If a cell does not have centrioles, what can it not do?
Pull apart the DNA fibers to divide.
Yes, Is that what you're going to say? Yeah, divide. If a cell does not have centrioles, it cannot divide. So, can neurons divide? No. Then we have the nucleus, okay? This is where we have the DNA and RNA. It's not really different than other cells. The nucleolus resides within the nucleus. This is where RNA synthesis happens. Synthesis, just a fancy word for we are making something. Nissl bodies, we can see Nissl bodies that are clustered and sort of situated around the nucleus. Sorry, my throat still hasn't recovered from when I had that cold. So the Nissl bodies that you can see here are clusters of rough ER. So if there's clusters of rough ER and ribosomes, what is produced in those areas? Does anyone remember? I see one person with an idea. Did you want to go for it, Harper?
Protein?
Yeah, protein. Is that what you're going to say? Awesome. Very good. Protein.
Oh, right.
And then the axon hillock. So we have this rounded cell body, and then it starts to kind of narrow out a little bit. This is the axon hillock. This is where the action potential begins. So the whole purpose of this neuron is to be able to conduct electrical impulses, to send action potentials. This is the job of the neuron. So these action potentials start at the axon hillock, and then they travel down the axon. You have heard this word before. Last week when we looked at how muscles contract, we said something along the lines of the action potential travels down the axon of the motor neuron. So now we can see the axon is really, it's just the little highway that it travels down on the neuron. The length of the axon will change dramatically depending on how far down we're going. So if we're innervating some muscles of the lower leg or the toes, that's going to be a long axon because it's much farther from the spinal cord. The synaptic terminals. So you've heard this before, and variations of this terminal. Like when you're at an airport and you go to your terminal, that's the last place you go before you leave. So the terminal is the last place the action potential goes before it leaves the neuron. It's the end. After the synaptic terminals, that action potential passes to whatever the next cell is. So in this case, it's a muscle fiber. And then dendrites. We're looking now on the opposite end of the neuron. The dendrites are these branches up here. Dendrites receive information. So the synapse is where the action potential is sent off. Dendrites are where we are receiving information from. Now, if we're traveling down the axon, we might have another little pathway that we can take. That's called a collateral branch. Collateral branch is just a branch off the axon that will eventually end in its own synaptic terminals. It's just a really nice, efficient way to have a message sent out to more than one location. Okay, there are 3 different structures of neurons. multipolar, which is the most common, unipolar, and bipolar. So multipolar here, this is the type that we were just looking at in all those pictures. These multipolar neurons are motor neurons. So the types of neurons that send an action potential that innervates a muscle fiber are multipolar. What makes it multipolar? A multipolar neuron has the cell body up at the top with multiple dendrites coming in, and then one single axon that ends in the synaptic terminal. These, as I was just saying, can vary in length greatly depending on where they're connected to. So when you wiggle your toes, that signal has to travel, branches off from the spinal cord, it travels down the axon of the motor neuron, travels all the way down until it gets to the right muscles. So the length of that motor neuron is going to be quite long compared to if we were to, even just compared to wiggling your fingers, the distance between the spinal cord and the fingers is not as long as the spinal cord to the toes. Does that make sense? Unipolar neurons, physically different than multipolar. We can see that the cell body is kind of hanging off to the side here, but it does not physically divide the dendrites from the axon. So here's the dendrites, the receiving end. Here's the axon, cell body's hanging off to the side, and then the synaptic terminals at the end. These unipolar neurons are what our sensory neurons are like. Our sensory neurons are the types of nerve cells that receive the information from our environment. So someone behind you is trying to get your attention. They very lightly tap you on the shoulder. You feel that tapping sensation. You can feel them brush up against your hair as they move to tap your shoulder. You feel the pressure that they're placing on your shoulder. That sensory information is received at the dendrites, travels down the length of the axon, and goes to the synaptic terminals. These can also be quite long. If you step on something, you can feel that sensation of that thing you stepped on. That is also being received at the foot. That information still has to travel up the leg and to the spinal cord, so that can be very long as well. And then we have bipolar. Bipolar neurons have one dendrite, one axon, and then a cell body in the middle. Bipolar neurons we see as interneurons, which we'll talk about today. They're basically connector pieces that sit between sensory and motor neurons. And we also see them in the special sense organs. We will do a whole section at the end of the semester on special senses. Okay, 3 different functional classes of neurons: sensory, motor, and interneurons. So it's the 3 that we just talked about. Let's go through each one, starting with sensory neurons. So sensory neurons belong in the afferent division of the nervous system. They receive information from our external environment, take that information, travels through the peripheral nervous system, and connects to the central nervous system. When the sensory neurons deliver that information to the central nervous system, they are connecting a sensory receptor to the central nervous system. We have various sensory receptors for various sensations. And we'll look at this in a whole separate lecture, but we have sensations for touch, for pressure, for pain, for temperature. So lots of different types of sensory receptors. We can break sensory neurons into two different subgroups, somatic and visceral. Somatic sensory neurons are going to give us information about the external environment. This would be somebody tapping you on your shoulder. This would be the temperature of the room changing, anything happening around you, and proprioception. Does anyone know what proprioception is? That was fast.
Yeah, it's knowing where knowing where my hand is without actually looking at it.
Yeah, that's great. Yeah. Knowing where your body is in relation to the space around you. Yeah, very good. Proprioception. So when you're going down the stairs and you close your eyes and you can kind of feel with your foot, like, is there, not that you would go down the stairs with your eyes closed, but you know, that feeling of like, where is the next step? Proprioception. So those two sensations come from somatic sensory neurons. Sorry guys, my throat slipped. I just need a second. Been losing my voice like every night. I apologize. Then we have visceral sensory neurons that give us information about the internal environment. What pieces of information about the internal environment might we be talking about?
Maybe something like your stomach hurts.
Yeah. Yeah, sure. Pain sensations internally. Is that what you were going to say? Anything else?
Bladder and
Sure. That would actually be from a stretch receptor. Those are called baroreceptors, and we'll look at those specifically on another lecture. Yeah. So stretch receptors can tell us when our bladder's full. These are things, pain and stretch receptors, these are things that we are consciously aware of, even though they're happening internally. But we do have some things that happen internally that we are not consciously aware of, like the change in pH in our blood. So that would be an internal visceral receptor as well. Then we have motor neurons. Motor neurons belong to the efferent division. This is when we send information out. It's going to be some sort of response. That response has come from the central nervous system. So this would be activating our skeletal muscle, something that we're voluntarily controlling, or causing some involuntary muscle to work. There are two subdivisions of motor neurons, also same names, somatic and visceral. Somatic motor neurons are where we control our voluntary nervous system. This is our skeletal muscle. When we control things like our smooth muscle or our cardiac muscle, or we excrete things from glands, that is something that we do not have conscious control over, and that comes from visceral motor neurons. Then we have interneurons. Interneuron is a connection point between sensory and motor. This is how sensory neurons and motor neurons can communicate with each other. They sit in between. So a sensory neuron is bringing sensory information to the central nervous system. That sensory neuron is going to connect to an interneuron. The interneuron is the connection to the central nervous system. and then connects to a motor neuron to get the signal sent out. Yes?
So an interneuron is like a telephone wire?
It connects sensory and motor. Okay, the last section that we're going to look at today is just naming some components of the central nervous system. I want to say, I think every new term I have listed on here is also on your terms list for lab. So the central nervous system, again, is the brain and the spinal cord. Very important, very delicate structures. So we, of course, protect the brain and the spinal cord by placing them in strong bones, right? The skull and the vertebral column. But beyond that, we have protective layers that sit on top of the brain and the spinal cord. It's not just the bones straight on top of them. We have protective layers. These protective layers are called meninges. Meninges. If you've heard of meningitis, itis is inflammation. Meningitis is inflammation of the meninges. The meninges are 3 layers that wrap around the brain and the spinal cord. The outermost layer, or most superficial layer, is the toughest layer, the thickest layer. This is there primarily for protection. This is called the dura mater. This is what the bone will sit directly on top of, the dura mater. Then we have a middle layer called the arachnoid. The arachnoid is there to help reduce friction and any rubbing between the layers. And then we have the innermost layer. This is the thinnest layer. This contains blood vessels, and this is what hugs the brain and the spinal cord the closest. Oh, so this is a picture of what they look like, the meninges surrounding the brain. But we also have them surrounding the spinal cord. It takes a second to realize what you're looking at here. We have a model that looks just like this in lab, so you will get used to seeing this kind of view. What we're looking at here is a vertebrae. This is the spinous process. This is the vertebral body. Here we have the transverse foramen. Is that starting to look a little familiar? It looks different than what we're used to because we have seen the vertebrae, but just by itself. Now we have the spinal cord stuck in the middle. So in the very center there, that is the spinal cord. We can also see these meninges wrapped around the spinal cord. If we were to take the spinal cord and kind of remove it from the layers, we would see the 3 layers here. Dura mater, arachnoid, then pia mater, hugging the spinal cord very close.
Yeah, just what the layers mean, because I was curious about the arachnoid. The other one is the dura mater, that is it's literally hard mother. The arachnoid is called that because it apparently looks like a spider web. And then tender mother is pia mater.
Well, that actually makes sense. Dura, hard, the outermost one. Arachnoid, I guess, just named for the appearance. The pia mater is very delicate, but hugs the brain and spinal cord the closest. Dura mater might make you think of the word epidural, which I think a lot of people are kind of familiar with that term, epidural. That is in reference to the epidural space, which you can see is filled with fat in this picture up here. And that's where epidural is like the spinal block where medication is delivered. Okay, so the spinal cord. The spinal cord is where the central nervous system continues down from the brain. So the brainstem transitions into the spinal cord. This is, of course, protected by the vertebral column. But most people don't realize the spinal cord actually ends at the beginning of the lumbar vertebrae. It doesn't just stop, though. I mean, we have branches that are coming down after that. So the spinal cord is like one unit up until we reach the lumbar vertebrae, and after that, it's just branches, many, many branches. This section here is called the cauda equina. Cauda equina. Equina, like equestrian, which is in reference to like horses, horseback riding, and that's named because it looks like a horse's tail. So the cauda equina. After the spinal cord is just many, many branches. In addition to the cauda equina, the spinal cord has two areas that the cord itself is enlarged a little bit. This is the cervical enlargement and the lumbar enlargement. This is where we have a lot of nerves branching out. In the cervical enlargement, we have many nerves that are branching out that innervate the shoulders and the arms. And the lumbar enlargement is where we have innervations coming out that innervate the pelvis and the lower limbs. And all it is really is just an area of the spinal cord that's just a little bit bigger. Okay, in the very, very center of the spinal cord, we have what's called the central canal. And then we have kind of an inward folding of the spinal cord on the anterior and posterior side. So the anterior median fissure, on most pictures and on the models that you'll see in lab, this really just looks like a line. And same thing with the posterior median sulcus. You remember those words, fissure and sulcus, from the skeletal system. This is just because of the way the spinal cord developed. We have this little inward folding, and those are the names of those spots. Okay, the dorsal root ganglion is this little cluster here, this ball, and then the dorsal root, and then ventral root's on the next side. So dorsal root ganglion and dorsal root here. Remember at the beginning of the semester, we were looking at anatomical terms. Anterior, posterior, superior, inferior. We had the terms dorsal and ventral. And I was saying that for the most part, we don't even use these. Okay, well, for whatever reason, we do use these in the nervous system. The way we use them, though, are synonymous with anterior and posterior. So dorsal. Dorsal is synonymous with posterior. So the dorsal root ganglion and the dorsal root are coming out of the spinal cord on the posterior side. The dorsal root ganglion is a cluster of sensory neurons, and the dorsal root itself is the axons. So what is happening on the dorsal root and the dorsal root ganglion? If it's a cluster of sensory neurons and axons, this is where the sensory information comes in and physically connects to the spinal cord. So sensory information comes in from sensory neurons and connects to the spinal cord with the dorsal root ganglion and the dorsal root. In a similar way, we have ventral roots. Ventral roots are the axons of motor neurons. So when you are going to move your body in some way, that information is sent from the brain down the spinal cord and then out to a motor neuron. It leaves the spinal cord through the ventral root. And that's the pathway it takes. Now, if you look closely, the dorsal root and the ventral root If we travel down, we'll see that they actually combine. They combine into what we call a spinal nerve. Spinal nerves are considered mixed nerves. They are mixed nerves because they're mixed with both the motor neurons and the sensory neurons. So we travel the same spinal nerve. But as we get very close to the spinal cord, the motor information goes on the ventral side, and the sensory information goes on the dorsal side. In the very center of the spinal cord, do you see this gray matter here? It's called gray matter. I think it looks like slightly brown, but for whatever reason, it's called gray matter. And you can see it in the very center of the spinal cord here, the gray matter. This is cell bodies of neurons. The gray matter forms little projections, which we call horns. So I think it's actually clear to see in this top picture. So here's the gray matter, and you can see that there's a peak right there, there's a horn. So there's one on the posterior side, one on the lateral side, and one on the anterior side. The posterior gray horns are where we have sensory nuclei. The anterior gray horns, we have information for controlling skeletal muscle. And the lateral gray horns here is where we control smooth muscle, cardiac muscle. As far as referencing goes and remembering your directions, I like to remind myself that this right here is the spinous process. The spinous process is what you can feel when you run your hand along your back. So is the spinous process going to be the anterior or the posterior side? Posterior. So anything that's closer to the spinous process is posterior. Therefore, this gray horn here is posterior. We're almost done. Then, if you're looking again at the spinal cord, we have all the gray matter in the middle, but all the stuff around it is white matter. So all of this here, and here, that's white matter. Mostly what we're seeing in white matter is axons, and these axons are either myelinated or unmyelinated. A myelinated axon just means that it has a protective coating, a protective wrapping around it called a myelin sheath. It is the myelinated axons that give the white matter its color. The white matter we can divide into different sections. So if we were to look at just the space here in between the posterior gray horns, that would be the posterior white matter. Then this chunk on the side here would be the lateral white column. And this chunk here would be anterior white column. And this is going to be a mix of either sensory or motor information. I feel like that might even be my last slide. It is my last slide. Questions on this? Let me stop recording here.